many babies 3-NIRS. And so you will see represented many of the general many babies principles, and this being sort of for now the only one brain imaging many babies project. There are also some specifics about it, and so I'm not going to be giving the talk. My name is Judith, whichever way you pronounce it. I'm the lead on the project, but luckily for me, we have Xin Sun actually, who is going to present the project today. She's a postdoctoral researcher currently working in Janet Worker's lab at UBC. And Xin, whenever you're ready, I think you can get started. One practical thing, if just keep your mics off for the talk. If you have questions, either put them in the chat. We're going to be monitoring that, or keep it to the end for the end of the talk if it's a real discussion question. If it's a minor clarification question, use the raise hand thing, and then we will let you ask your clarification question. So chat, clarification questions during the talk, and substantial questions at the end. Okay, here we go then.
Okay, cool. Can you guys hear me? I believe so. Okay. Thank you, Haiti, for setting us up, and thank you. I actually Googled your name like you did, right? Thank you, Judith, for doing the introduction. All right, so today I'm gonna present on behalf of the Many Babies 3-NIRS Consortium, and as you can see that joining us today, we have Judith, who is our principal investigator for this project. And my presentation is a large-scale multi-lab replication study assessing infants' ability to detect irregularities in speech. So I believe the Many Babies initiative, an important driving factor would be because of the replication crisis. So it is the phenomenon that many empirical studies, like in psychology and medicine, like in other domains, they do not replicate. So it is certainly concerning, but to me, it also raised our awareness of we need to look in greater detail in how we do research and do something to fight against replication crisis. So what's the driving force of replication crisis? Why there's this thing happening? So a survey that asked thousands of researchers this question, what's the factor that contribute to these irreproducible research? And there are certainly factors reported, like academia is so competitive, we have so much pressure to publish. And there are also factors around the research themselves. For example, a lot of research, they were hard, or they were challenged, they were themselves challenging for other people to replicate, they don't put up their methods or codes or raw data for people to do reanalysis or replication. And others might be around the original study is flawed in different ways. For example, it lacks statistical power. It itself is poor in its experimental design or analysis, and so on. So now we sort of get a sense of how researchers think of replication crisis and the driving force. What could we do? So what's the solution here? So there are a lot of things that we could do. One of it is to conduct open science practices, and this is recent years is very hard, and a lot of people are doing this. So for example, we could do pre-registration before we conduct the study, or before we collect data, we pre-register our hypothesis. And we could put up our data online so other people can access to it to conduct replication or reanalysis. And a lot of journals, they're open for register reports, so you don't have much of a publication pressure before, I have to report the significant results before you collect data, basically. And another thing we could do is to conduct meta-analysis. This would significantly improve our statistical power in individual studies. And a good practice would be also to include unpublished studies, which are very likely to have null results. And what else we could do is to conduct these multi-lab replication projects. It has all the benefits of the previous practices. And there are certainly a lot of initiatives doing that, like many labs and the many babies that I'm going to talk about today. So what's many babies? Many Babies initiative is aimed to conduct multi-lab replications of key studies in developmental psychology. And we had a lot of projects already published, like Many Babies 1, and so a lot of them, they're ongoing. And today I'm gonna talk more about Many Babies 3-NIRS project. So what is our project? Our project is aims at replicating an artificial grammar learning study among infants. And this study was first published in 2008 by UDIT here. And for our specific, like Many Babies study, we aim to collect data from all over the world, well, not all over the world, but like 31 labs all over the world. They are from like 12 countries, 4 continents. And like UDIT has introduced, I'm now sitting at UBC, and our lab will be proudly contributing to one of the samples. All right, so here's our timeline. So UDIT has this first idea back then from like 2020. So the idea was for sure to replicate one of this classic study. So what's the classic study is about? So what's the UDIT 2008 study is about? So this study was published in PNAS, and the title was "The Neonate's Brain Detects Speech Structure." So in this study, they recruited 22 newborns. Their ages are from 1 to 6 days of age. They're super young. And what the study did was to let these babies hear a series of sound stimuli. And what's the sound stimuli? So it has 2 kinds of sound stimuli, and they represent 2 kinds of artificial grammars, either ABB or ABC. So like you can see, the ABB sequence, the second syllable and the third syllable, they are identical. So like here, these are all these examples. And as for ABC sequence, these 3 syllables, they have more diversity. They are all different from each other. So basically, the newborns, they hear either ABB sequence or ABC sequence. And the study design followed a block design. There were ABB blocks and ABC blocks, and in between different blocks, there are silence periods. And within each block, there were 10 trials of different either ABB or ABC stimuli. And while the newborns, they were hearing these sound stimuli, their brain activities were also recorded through the FNIRS neuroimaging technique. So what is FNIRS? FNIRS 101 here. So FNIRS stands for Functional Near-Infrared Spectroscopy. It is an optical neuroimaging technique. So you can see here, this kid is wearing a FNIRS cap, and there are a lot of optodes on her head. And these optodes can either be light sources or light detectors. So for light sources, well, it's straightforward that they shed lights into the tissue. And for light detectors, they collect lights from the tissue, coming outside of the tissue. And here, changes in lights between these 2 like optodes would reflect changes in the blood flow in this particular brain region. And therefore we got to know, okay, so while the kid is working on something, this brain region is super active, and then it will have changed its blood flow, and then from the FNIRS light changes in lights, we could know like, oh, okay, it's this region that are engaged in this this task. All right, so this is the end of FNIRS 101. Let's go back to the original paper and look at how their babies were measured. So their babies also wear a FNIRS cap, and the brain regions that are covered are here. So basically they more focus on the frontal, temporal, and parietal regions, and their most important ROIs, regions of interests, are centered around the frontal sides, which are these pinky circles, as well as the temporal sides, which are the blue circles. Yeah. And because like for a simple reason why these are the ROIs, because like for temporal regions, these are auditory regions, and the frontal sides are more associated with language processing later in life, and they're hearing something, right? So it is obvious. And what they found. So what did they find? So let me direct you first to the legends here. So I want to direct you first to this pinky dotted line here. This line represents the ABB condition, the brain activation for the ABB condition. And this red line would represent the brain activities for ABC condition. So if you look at, for example, channel 5, we can easily see that the pink line is quite different from the red line. And indeed, so they were like in these left frontal sides, as well as the bilateral temporal sides, the pinky line was significantly different from the red line. What does that mean? That would mean that, well, when the babies hear the ABB sequence, their brain were more sensitive by showing enhanced brain activations in these left frontal as well as bilateral temporal regions, as compared to when they hear the ABC sequence. All right, so then a closer look, like a more 3D view of what are the regions that are activated, that are significantly different between ABB and BBC. We can easily detect that these are located in the left frontal regions as well as the bilateral temporal sides. And what does this study tell us? Well, in summary, newborns, they were able to discriminate simple structures based on immediate repetitions like ABB here. And this is like the study that we want to replicate here. So so here's our idea and our like study. And going back to our timeline, where are we now? We are here in the preparation stage. Now we are trying to set up the study design, the stimuli, and plan for later data analysis and acquisition and everything, basically. All right, so what's the plan? We aim to recruit participants ages from 0 to 24 months. We had a relatively flexible age range here for several reasons. First, we have so many participating labs, and not all the labs, they have access to like newborns. And second, and importantly, it'll be very interesting to also look into how babies, when they grow, as they grow up, their brain reacts to these sound stimuli, basically drawing out the developmental trajectory of babies' neural mechanisms for these sound sequences. And the participants will be around 40 per sample, typically developing kids. And this sample size was calculated from a power analysis here. All right, so then moving on to stimuli and paradigm. We'll use we'll maximally match the stimuli used in the previous study. We'll again use the ABB and ABC grammar conditions, and we'll use synthesized stimuli. And now it is already created. They're already created by one of our core members, Irene. Unfortunately, she couldn't be here, but yeah, she did a lot of work. And as for the study design, will still follow a block design. We will include ABB blocks, ABC blocks, and we'll also include a mixed block with both kinds of stimuli. We'll see what would happen here. All right, then when moving on to data acquisition, we hope that the labs across the countries or sites, they will use their general experimental protocol as well as their NIRS machine. They may come from different NIRS companies, but it's totally good. And but we will make sure that their caps would target temporal and frontal sites, which are the regions of interest from the original study. And then when it comes to data analysis, all right, I see yeah, I guess I'll leave the question later. All right, so when it goes to data analysis stage, now we have built our little teams in responsible for different aspects of data analysis, including brain localization, and data quality assessment, and building a criteria for data inclusion, how to remove say the pre-processing stage, remove motion artifacts, things like that. And then we'll when it goes to the formal data analysis stage, we'll use different kinds of methods like mixed effect modeling as well as meta-analytic procedures. And from these statistical analysis, what we aim to have, for sure, first we want to know the general effect size of the study design. Basically, would we replicate the original study? And the other thing is, well, there might be a lot of variations, say across labs or across different potential moderators. We want to know what are these moderators. For example, if it's age that drives some effect size differences, or say sites or culture, things like that. And I see the chat that it might be different languages, right? And another big thing that we expect to do is to bring together the NIRS researchers and have a conversation about how to better apply NIRS to baby research, as FNIRS is a relatively new imaging technique, and a lot of labs, they're trying to figure out their own way, and they have their own protocols of running NIRS studies. And by our NIRS, Many Babies 3 NIRS project, we hope to bring together these researchers, have a conversation, and do a study together, hoping to move forward the domain, basically. All right, so here's our plan. And going back to our timeline, moving forward, we hope to do a lot of open science practices. So we hope to submit a registered report early next year, and we hope we're also doing some other practices, like we already set up our OSF page, we put up already our stimuli and drafts and stuff, and we also had our GitHub page. If you're interested, feel free to check it out. Yeah, so that's my presentation. Thank you so much. Big thank you. And yeah, now we're open for questions, and Judith would join me to answer all these questions too. Yeah, we have 10 minutes, I believe.
Yeah, so maybe we can start with the question in the chat.
Mm-hmm.
I don't know if maybe I'll take this one because I think you did not join the project at the time we developed the stimuli. So,
Yeah.
right. So the phoneme repertoire was developed on the basis of empirical, first aimed to narrow down the phonemes that we use to a universal set. That is, some phonemes are more common in the words languages than others. So we chose stop consonants, for example, are fairly common, or at least certain stop consonants are fairly common. Among vowels, there are also some that are very common. So we aim to use those that are most common. And of course, this will not cover everything. So certainly we will use some phonemes that in some of the languages we will have are not phonemic, are not contrastive. That in a certain sense is not necessarily a problem because we are not expecting these babies to identify the phoneme or even the syllable as such, as long as they can recognize that it's the same thing. So if one child hears a syllable as ta, ta, and the other as da, da, we don't mind as long as they hear them as identical in the repetition condition, and of course all different in the other condition. And so we made sure that the A, B, and C syllable categories are maximally distinct. So they were like, this is built into the stimuli. So that's roughly it. And then we used Ambrola, so it's a synthesized sound. And there we chose a voice. So Ambrola has voices, meaning phoneme sets for different languages. And we just ran a survey with adults, so not babies, from the labs that were involved at that point in the project, asking people to evaluate the sounds for quality, cleanliness, distinctiveness, etc., etc. And we came up with, I think, Turkish one or something. I forgot exactly which phoneme set we came up with now, but this was based on an empirical survey.
Cool. I also get to know more.
Okay, so another question, right, the age question, right? So the original study was conduct newborns, but there's now submitted, hopefully soon published results from my lab suggesting that by 6 months of age, the responses to the ABC, so the diversity condition, changes. And there is also older evidence from Chuck Nelson's lab suggesting the same at 7 and 9 months. So there is reason to believe that there is an interesting developmental trajectory. And so we are looking, and that has theoretical consequences about how abstract these representations are. And so we are interested to document that. And as Xin also told you, it's simply also just practical. Namely, some labs have access to certain age ranges, but not others. You can now also unmute. So those of you that are in an environment where you can talk, you can unmute your, so raise your hand and then unmute your mic and ask your question vocally if you prefer that. But of course, we are still monitoring the chat.
Yeah, if you have any suggestions and things, I mean, this is the first neuroimaging multi-lab, like many babies project. So any suggestions, recommendations, be welcome.
How people are thinking, just going back to what we said in the presentation, indeed NIRS is relatively new, and importantly, practices are not standardized. Here variability is much greater, and it requires, So data analysis, like this is highly dimensional data, right? So data analysis has a lot more of these somewhat arbitrary experimental degrees of freedom in terms of decisions on filtering, pre-processing, all of these things. And so in a certain sense, we're really hoping for the developmental community as well as for the NIRS community in general to arrive at some kind of standardization or at least recommendations for practices based on empirical data. So the project in a certain sense includes sort of mini projects or mini sort of experiments of its own, just looking at what would be the best way to filter the data, what's the best way to remove artifacts or correct for artifacts, and a number of these decisions that are very common in imaging and much less common or much more limited in behavioral data. So this is a huge challenge in this project. Any other questions in the meantime?
Yeah, thank you all.
Just like one maybe last comment is, of course, we are still not even at the submission stage of the stage one report. So anybody who is interested is, of course, more than welcome to join. One thing that one question for the NIRS machine or expertise with NIRS to join. And I would like to point out that this is not the case, right? So it is okay to join even if you won't be able to test, won't be testing babies, or you won't be able to test babies. There's a lot to do from write-up to presentations to data analysis where you don't really have to have a NIRS machine to join this project. So this is to encourage everybody, even if you're not a NIRS expert, you've never done this, you don't have a machine, to consider joining.